Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to be focusing on viruses. So what are viruses? Viruses are obligate intracellular parasites, so they are only able or obligated to reproduce within/intra cells. While within the cells, viruses have some attributes of living organisms, such as the ability to reproduce. But outside cells, viruses do not carry out metabolism and do not have the cellular machinery needed for reproduction or energy production. Overall, a virus is nothing more than a package of nucleic acid that says pick me up and reproduce me. So let's talk about the basic viral structure. Starting with the capsid. The capsid is a protein coat that surrounds the viral genome. The bacteriophage capsid consists of a head, where the nucleic acid is, and a tail. The tail fibers and base plate attach to the host cell, and the central shaft, also known as a sheath, contracts using the energy of stored ATP and it injects the genome into the host. The bacteriophage has to inject its genome into the host cell, as it must puncture the bacteria's cell wall. Animal cell viruses do not have to do this, as they can be internalized whole into animal cells because they do not have cell walls. Now let's talk about the envelope. A lipid bilayer derived from the host cell membrane. Not all viruses have this. This is found in most animal viruses, and this is a membrane on the exterior of the virus and is derived from the host membrane. This can hide the viral capsid antigens, which are normally recognized by the host's immune system. Viruses that don't have envelopes are called naked viruses. All phages and plant viruses are naked because viruses gain envelopes by budding through host membranes. But when phages and plant viruses exit the cell wall is destroyed and host membranes rupture, leaving no membrane through which the remaining viruses must bud. Now let's talk about genetic material. Genetic material can be DNA or RNA, single-stranded or double-stranded, and is either linear or circular. However, a given type of virus can only have one type of nucleic acid as its genome, and a mature virus does not contain nucleic acid other than its genome. A factor that influences all viral genomes is size as a limiting factor. Viruses are much smaller than the hosts they infect. An average virus is between 100 to 200 nanometers, whereas an E. coli bacteria is between 1 to 2 microns, and a small eukaryotic cell is between 10 to 20 microns, and 1 micron is 1,000 nanometers. Viruses come in many genomic varieties, like I said before, but the genome type determines the replication strategy. Now, if they're double-stranded DNA viruses, like adenoviruses, they use host polymerases to replicate and transcribe. If they're single-stranded DNA viruses, they must first be converted to double-stranded DNA. And of single-stranded DNA viruses, you can have positive sense and negative sense RNA. Positive sense RNA can be directly translated by host ribosomes. Negative sense RNAs require RNA-dependent RNA polymerase to synthesize a complementary positive sense RNA strand. They must carry an RNA replicase in the virion to ensure that the complementary strand is synthesized. Finally, you have retroviruses. The example of this is HIV. These have positive single-stranded RNA and use reverse transcriptase to make DNA. This integrates into the host genome as if it's a part of it. This allows for the cell to be infected indefinitely, and the only way to cure this is to kill the infected cell itself. This is why HIV is so difficult to treat. Now, let's talk about the general viral replication cycle. So first step is attachment or adsorption. This is spelt as the same way as absorption, but instead of the B, it's a D. In this, the virus binds to specific receptors on host cell. After this, you have penetration or eclipse. This is where you enter via endocytosis, membrane fusion, or injection. After this, you have uncoating, where the capsid breaks down and releases viral genome. Then you have replication and protein synthesis, which varies depending on genome type. Then you have assembly, where new virions are put together. And finally, you have release via lysis or budding. First, let's talk about bacteriophage life cycles. So here, after penetration or eclipse, The bacteriophage can either enter into the lytic or lysogenic life cycle. The lytic cycle is where the virus takes over host machinery, replicates, and makes genes. The first gene product it makes is hydrolase. This degrades the entire host genome. Hydrolase is an example of an early gene. Early genes are genes that are expressed immediately after infection. Then, multiple copies of the phage are produced as well as capsid proteins. For this, each capsid automatically assembles around a new genome. Finally, an enzyme called lysozyme is produced. This is a late gene. It destroys the bacterial cell wall, and since osmotic pressure is no longer counteracted by the cell wall, the bacteria lyses, releasing about 100 progeny viruses called virions. These virions then affect other bacteria. Bacteria in the lytic phase are termed virulent. The lytic lifestyle is a good way to rapidly increase numbers, but it destroys host cells, which is a problem. Some viruses are cleverer and enter the lysogenic life cycle. In this life cycle, the viral genome integrates into the host genome and is called a prophage. Here it replicates with host DNA. The host is now called a lysogen. The prophage is silent and does not express its genes. This is because it is blocked. But the cleverness of this life cycle is that every time the host cell reproduces, so does the prophage. Eventually, the prophage activates and removes itself from the host genome in a process called excision and enters the lytic cycle. You may recall last episode where we were talking about transduction, and here the host DNA may accidentally be packed into a viral capsid. And this happens in the lysogenic life cycle when you are excising from the host genome. Now, the animal virus has the same lytic life cycle as the phage, but it also has a productive cycle. This is similar to the lytic cycle, but it does not destroy the cell. When the virus leaves, it can leave by budding, so this does not destroy the cell as the membrane can reseal. The lysogenic life cycle is similar, but the virus is called a provirus instead of a prophage, and this stays dormant for an indefinite period of time until one day, usually when the host encounters stress, which could be from a lack of sleep or preparing for a very important entrance exam, the virus reactivates. I just want to quickly touch on prions and viroids. Prions are infectious proteins that cause disease by triggering misfolding of other proteins, usually involving the conversion of a protein from an alpha helical structure to a beta sheet. This drastically reduces solubility of the protein, and eventually the protein aggregates, and this causes the form and function of the cell to be reduced. Prions are known to cause mad cow disease, Creutzfeldt-Jakob disease, and familial fatal insomnia. Viroids are small pathogens consisting of very short, circular single-stranded RNA that infect plants. Viroids can bind to a large number of RNA sequences and will silence genes in the plant genome. This prevents synthesis of necessary proteins and can cause metabolic and structural derangements in the plant cell. Viroids are plant pathogens, but there are a few human viroids that exist, including the hepatitis D virus, or HDV. HDV is innocuous on its own, but when co-infected with hepatitis B virus, or HBV, HDV can exert its silencing function on human liver cells. Now that's it for today. In the next one, we'll go over some microbiology practice questions.